Chris.
Hi.
Hi. Okay, so we're coming to the end of this session, but there's just a couple more things to do. If you do have any questions about any of the talks or anything you want to bring up or anything to discuss, please do put it in the chat. A bit quieter this afternoon than we were this morning. We've had lots of questions this morning. So there's not a great deal more to talk about, but one thing to mention to you is that we have put together some homework. You think, oh, homework, that sounds dreadfully boring, but it's really to try and help consolidate your knowledge, you know. So it's there if you found some of the talks interesting and you want to learn more and you want to explore it a bit more. Some of the talks from this morning, this afternoon and tomorrow have homework assignments associated with them. We're not going to be marking you or anything like that, but they are there to do. And if you want to discuss them, that is what the Slack is there for. And of course, the Slack is there to continue conversations and to ask other questions that might occur to you that have arisen from today to any of the instructors, and a few of us are on there. So you can also discuss the homework on the Slack if that's something that you're interested in. So I just wanted to let you know about the various pieces of homework that are there and what they cover. So the first, this is on the course website, so you'll have this link because this was sent out to you when you registered. The first piece of homework is from Josh. It's covering, in this case, the primal scheme web interface. And you'll remember the primal scheme web interface is how you can generate your own amplicon schemes for either for sequencing SARS-CoV-2, but actually more generally for sequencing any RNA viruses. And so the homework link is a Google Doc, and if you click on that, it really gives you a walkthrough on how you can design your own scheme. And this is based around Ebola virus. It could be based around any virus, but here you start with some example Ebola reference genomes, and it goes through the steps for creating your own primal scheme. So you can do that if you're interested. Another piece of homework related to the Arctic bioinformatics pipeline, and this specifically relates to the Oxford Nanopore version of the pipeline. And this tutorial was prepared by Will. And this particular tutorial is done in the form of a binder notebook. Now, I must admit, I haven't tried this out for a while, so I don't know whether this is still working. Hopefully it will still work well. But if you click on the binder notebook, you can you get this kind of worked example, which is a tutorial about how to install and run the pipeline, but is also live. So in theory, you can run the commands that are specified in this notebook. So after this is already installed, and then you can actually run the commands from within the web browser. So if you click on this, in this first cell, it's just running the Arctic script just to get the version number, and you can click the run button, and in theory, that comes and that runs and returns the results. I think it did work. Yeah, so it's actually running this. So if you follow these instructions, it shows you various different commands in the Arctic pipeline that you can run, how to download a primer scheme, check that the scheme is valid, look at the primer scheme, and then run it on some real data that we've supplied, see what the output is, and see what the files and the QC that are generated are, and then look at it. So you can just run through this, very easy to see how that pipeline works with some example data. There is some homework associated with Nabil's tutorial presentation on bioinformatics gotchas. So this is looking at things that can go wrong, focusing a little bit on sample identifiers and metadata errors. There's just a few questions there. It's quite an easy and fun one to do, and the answers are listed at the bottom. We haven't covered this yet, but tomorrow we're going much more into the evolutionary and phylogenetic analysis of sequence data. So after we've gone through the process of generating sequence data, we've gone through the process of converting raw sequence files into consensus sequences, and then we've talked about various ways of checking that the data is good quality. Now you actually want to do some analysis. We'll cover that tomorrow with talks from Verity, JT, and the usual talk from Anya will be given by Rachel tomorrow, and she'll talk about the pangolin software and how we assign lineages with pangolin. So there is a homework assignment, which is a web page for how you can use pangolin, and this is a web version of pangolin that actually runs on Climb that you can play with. There is some basic description of how you install Bioconda, just in case you want to do that and need to do that to install packages. There is some homework around our system called Lab on an SSD. We haven't got a presentation about that this time, but this Lab on an SSD is a way of rapidly installing a Linux environment on your laptop. So if you use a Windows laptop, particularly, possibly a Mac, an older Mac usually, there is a way of booting up your laptop into Linux and have many of the software packages that I've talked about, Arctic Pipeline, the mappers, etc., already installed on a process called Lab on an SSD. To use that, you'll need either a large memory stick, at least 32 gigs or 64 gigs, or a solid state drive, typically 256 gigs or larger. And you can install, if you've got a spare one that this will remove the information on that, so don't use an important one, but you can install a bootable Linux system on there. So that's the instructions for how to do that, something we maintain in the Arctic and Climb projects. And then finally, another tool that will be introduced by Rachel tomorrow is Civit. Civit is a tool for producing trees and doing genomic epidemiology on clusters rapidly by looking at a set of sequences that you've produced and putting that in the context of all of the available sequences in the public databases. So Civit will be talked about tomorrow, but there is a tutorial. If you click on Civit tutorial 1, that will download a Word document, and that gives you the instructions for how to download, install, and run Civit. So there's plenty to get on with, and obviously you don't have to do any of these, or you may want to just do a few of them, the ones that are most related to your particular interests and work, but those are there, and these links are persistent. They'll be there indefinitely to do in the future if you want to. Okay, so tomorrow we start again at 9:30 GMT. So we have really good talks on genomic epidemiology, introduction to genomic epi, introduction to phylogenetics, a basic guide to lineages, variants, and how to assign lineages and variants, and then finally, how do we detect and discover and characterize novel variants. All of those talks are fantastic. So I hope to see you tomorrow. But of course, if you're not available because of the time zone or because you're busy, the recordings will be posted on the website as soon as we possibly can get them up there after the course finishes, and they'll be there permanently. So that hopefully answers some of the questions in the chat. As Lisa, thanks to Lisa and Mavis for helping organize this course. Lisa is just pointing out that if you need a certificate of your attendance for the course, as many people do, we will provide you one, but only at the end of the course and only after submission of some feedback. So we'll give you the feedback link tomorrow, and it will also be published on the web page for you to look at. I think that covers everything. So we'll thank you everyone for your attendance and your questions today. I hope you found the first day useful and stimulating, and I certainly hope to see everyone tomorrow for the session. So thank you very much. Thanks for your feedback in the chat, and I hope you have a great rest of the day, whatever time it is in your time zone, see you soon. Gustavus just asked one question just there. Can we use the homework to install the last version of Arctic? Well, that homework will install the current stable version, which is version 1.2.1 for the nanopore pipeline. We don't currently have any homework for the next flow pipeline, which is more what Sam talked about this morning. We will look into generating some homework for that pipeline. I can't promise it will be available today, but we'll look into doing that in the future. Hope that answers that question. All right, thank you all for your attention. Thank you very, very much to all the speakers today, and see you tomorrow.